Good morning, everyone. Welcome to the seminar series. We've got an exciting talk coming up today by Eric Caldera. Excited to welcome you all. There'll be a seminar next week as well on birds, so make sure to come back again next week also. But my task for this morning is to introduce Eric. Eric is currently a postdoctoral fellow in Paul Barber's lab. He's a president's postdoctoral fellow studying symbioses, which we're going to hear about. He previously did his PhD at UW-Madison and his undergrad work at UT Austin. So you've explored a range of latitudes in your scientific career and climates. And he said he loves symbioses and evolution and plays a little guitar on the side. I'm really excited to welcome Eric to tell us about your research. Thanks.
Good afternoon, everyone. Thank you for having me here at the EEB seminar. What I'm going to do today is talk about bacterial symbioses. I'm going to start with telling you about my doctoral research in fungus-farming ants. And I'll do a deeper dive and give you a more complete story with that. And then at the end, I'll tell you a little bit about what I'm working on right now involving bacterial symbionts in marine ecosystems. Okay, so coevolution, which we define as reciprocal adaptation, shapes the diversity of life, and symbiosis, which we simply define as living together or sharing an intimate relationship, promotes evolutionary innovation. And I would argue, there's no better example or extreme example of evolutionary innovation than the evolution of the eukaryotic cell, which emerged through symbiosis between bacteria and archaea. Another really cool system that is a great example of evolutionary innovation is that of the fungus-farming ant symbiosis. So what you're looking at right here is an example of a leafcutter ant. If you've ever been to the rainforest, you've probably seen these ants. They're arguably the most ubiquitous animal in the rainforest, in part because they cut fresh leaves and they form these long trails through the forest where you can see these green, fragments of leaves bobbing up and down as they're moving through the forest. Now, a lot of people see these trails of ants carrying leaves and they think that the ants are actually consuming the leaves as their food source, but that is not the case. The ants are doing is taking these little fragments of leaves, they go underground, and underground they have these fungal chambers or fungal gardens. They take the leaf fragments, they basically feed it to the fungus garden. The fungus garden then decomposes the leaves, and in return, the ants get these hyphal swellings and nutrient-rich gongolidia off of the fungus, and so the fungus serves as the primary food source for the ants. This system is indeed a true agricultural system in the way that we define agriculture. It is highly analogous to human agriculture in the sense that when we grow a crop, we have to deal with some of the negative aspects, such as pests and pathogens might infect our crop. So similarly, the ants have in their fungal garden, they have a crop pest that tends to live at the bottom, which is a microparasite, ascomycete in the genus Escovopsis. And similar to human agriculture, where we might spray an insecticide or a fungicide or apply something to limit this pest or parasite, the ants are doing the same thing. Growing on their cuticle, they have a symbiotic bacterium. This bacteria is producing antibiotics that are inhibiting the growth of the crop pest. So again, the example of extraordinary innovation through symbiosis. And what's even cooler about the complexity of this system is that it's not just in one species where this has evolved. There's a whole monophyletic clade of fungus-farming ants. The group is called the attines. And as you look over across the phylogeny of this approximately 250 species, you can actually loosely group the different genera into different agricultural systems where the ants are doing their agriculture a little bit differently. They're using different types of fungal crops. They're using different strains of bacteria, and also combating different strains of the Asclepius pathogen. So this is just an example of some of the co-cladogenesis across the ants, the cultivar, and the Asclepius pathogen. In addition, there's also specificity in the bacterial mutualist that they're using. The bacterial mutualist is in the genus Pseudocardia. It's an actinobacteria, and it's basically closely related to actinobacteria like Streptomyces, where we actually gain a lot of the antibiotics that we use in medicine for ourselves. So I'll point out that, I started with the leaf cutters because they're sort of ubiquitous and a lot of people know them. You see them on National Geographic. But throughout these, the attine phylogeny, the species that I focus on is the more basal group. Apterostigma dentidrum is the ant that I work with. And, unlike the fungus farming leaf cutters, where, they can have millions of workers, their colonies can literally grow so large that they can span a football field. And because of that, it's just hard to actually determine where one colony begins and where the other one ends. And if you're interested in working with the queen, you'll never find the queen unless you have a bulldozer or something. So these, in contrast, being more basal, their fungal garden is about the size of a baseball. And rather than cutting fresh leaves, they just sort of pick decaying plant material, sometimes caterpillar frass, and they use that to feed their fungus garden. So what I'm going to do right now is go through the different players that I've already told you about, but specific to the Apterostigma dentidrum lineage. And then moving forward, we'll sort of be concentrating mostly on this interaction between Pseudocardia and Asclepius. So just sort of remember, Pseudocardia is the bacterial mutualist, and Asclepius is the bad guy, the parasite or pathogen, if you will. So this relationship between the ant and the fungal cultivar, like I said, is an agricultural system. It's an obligate mutualism, meaning that the fungus cannot live without the ant and vice versa. Specifically within the agricultural system, the ants grow their crop in a monoculture. This partially results by the fact that the fungal symbiont is transmitted vertically, meaning that before a young queen takes off to form her new colony, she's going to basically bite off a chunk of her mom's fungus. She has a specific organ where she sort of keeps it in her throat. She starts her colony. She spits up this new fungal lineage, or continued fungal lineage. So that's how we get sort of clonality throughout the system. Now, you guys, if you know anything about agriculture, you know that there are trade-offs to having a monoculture or a clonal crop, meaning that you're going to be highly susceptible to disease because you're not sort of harboring the genetic variation to help you combat pathogens and such. Just kind of kicking me out here. Okay, so now I'll tell you about the dynamic between the fungal cultivar and the Asclepius pathogen. So it's a pathogenic infection. The mechanism by which the pathogen finds the fungus is through chemotaxis. So there's like a chemical attraction. As far as we know, this, unlike the cultivar, it's horizontally acquired, although we imagine there are probably opportunities to get some vertical transmission as well. From our experiments, we know it's primarily acquired horizontally through the environment, usually early on in the colony forming, again, through this sort of chemical attraction that the pathogen has. One thing I'll point out some results from an initial study I did in the system, where I looked at the population genetic structure of both the pathogen and the fungal cultivar. And basically, these are not congruent. So, if we imagine that the pathogen is also coming along vertically with the cultivar, we expect some population genetic structure congruence, but we don't actually see that. And so here's just an example of that. You don't need to stress the details, but basically we get, these sort of 6 main population clusters within the cultivar, and then we loosely get these 3 Escovopsis clusters, and they don't really overlap. In addition to doing the population genetic structure of these 2 symbionts, I ran these bioassays, and you probably have seen or heard of these before. They're just really old school microbiology assays where we grow up one microbe. In this case, we grow up the cultivar, we let it sort of sit there, exude its secondary metabolites, then we inoculate with the pathogen, and we see if the cultivar has the capacity to basically inhibit the growth of the pathogen, and we quantify whether or not there's a zone of inhibition there. This figure is a little bit boring in that we basically found almost no variation in that this specific strain of the pathogen can infect all of the cultivars, sort of regardless of the geographic region. In this case, it's throughout Central America, and I will sort of elaborate a little bit more on these populations. But I show this figure mainly to, again, articulate that kind of chemotaxis component, in that if you do these assays sort of across species or give them different cultivars, you'll find a lot of variation where in some cases the cultivars can really inhibit the pathogen and sometimes they can't. But this is just an example of how with this specific interaction, with this monoculture of highly similar genotypes in the cultivar, it really is highly susceptible to this fungal pathogen. This is basically example of what happens when the infection goes rampant within the garden. So this is a healthy fungus garden, again about the size of a baseball, and the ants are constantly dealing with this infection and keeping it at a low level, but basically there's sort of a critical point where if the infection gets too high, it can just completely overrun the colony. And this is basically what can happen to a colony in only like a matter of hours. So this could be a mature colony several years old, and it only takes a couple hours to completely destroy the fungal garden. Once the fungus garden's gone, the ants have no home and they'll basically die. So you can see how there's a really strong selection pressure imposed on all of the players in the system within. So now I'm going to tell you the specifics about the interaction between the pseudocardia and the Escovopsis. And this is basically kind of the main focus that main focus of my doctoral work is teasing apart this interaction. So we know that this association is ancient. In Dominican amber, tens of millions of years old, we can see the bacteria and the structure where it's housed on the ant, even within this specific genus. Again, it's characterized by antibiotic inhibition. The symbiont, the bacterial symbiont, is producing antibiotics. But some of the lingering questions here are, what is the transmission mechanism? And somewhat related to that question is, how is antibiotic potency maintained? So, I kind of mentioned before the trade-offs between vertical transmission and, clonality. So from a symbiont coevolution perspective, there's a benefit to vertical transmission. You know that your parent used this strain and it worked well. So there's security in having a strain that works well. But in contrast, that mechanism of verticality, is going to basically mean that you're going to have a highly clonal symbiont population, which again has its negatives. So again, how is antibiotic potency maintained over millions of years? You and I know that in human modern medicine, one of the biggest crises we're facing is antibiotic resistance, basically meaning that we're losing, the potency of antibiotics to treat disease. So, how are the ants maintaining this basically back and forth relationship over millions of years? And this is a graphic that just kind of illustrates, again, some of those ways that the transmission mechanisms could work. We can have vertical transmission of the pseudocardia. There's potential for transmission between colonies, although in our studies, we kind of don't think that's the case. They basically stick with the strain they form their colony with, and it's hard to get it to switch over. But alternatively, there could be another mechanism by which they're maintaining their antibiotic potency, and that could be through an acquisition hypothesis, where each generation there's a new founding of a colony, and the ants are basically taking up beneficial microbes from the soil and dealing with, using those beneficial microbes from the soil and basically maintaining more of a consortia of diverse bacteria. And, we see parallels in other systems. For example, in the old world, there are fungus-farming termites, and they do their system very differently there. Theirs is defined by horizontal acquisition, so they don't have a clonal crop. They go out and they forage in an acquisition model for their crop, and so each generation, and so they maintain a really diverse crop, and that's sort of one of the ways that they're combating disease. So just real quickly, most of the next figures I'll be showing are from some of my early work on the population structure and a recent paper that made the cover of AEM, and then also a genomics paper that's in review at the moment. So for my study sites, again, I was using Apterostigma dentigerum, and I was focusing on Central America, specifically in Costa Rica. Here, LS is La Selva Station, and also focusing primarily here in Panama, in the Panama Canal. And so the kind of focal populations here are going to be the three populations of BCI, which is Barrow, Colorado Island. It's a very famous research station. You may have heard of it. And it's effectively an island only because it was created during the flooding of the Panama Canal. So it's an island, it's about 100 years old. GAM right here stands for Gamboa Forest. It's a small patch of forest that's nested within a small city that used to house like the administration for building the canal. And along here we have what's called Pipeline Road, which is kind of a longer stretch that again used to serve to move things up and down during the building of the canal. As I mentioned before, the different genera, have different specificity in their symbionts. So with Apterostigma, they tend to have the actinobacteria concentrated under their first set of forelegs. And so, basically I spend a lot of time collecting these guys and then kind of a brute force effort in trying to get these microbes into culture. I'll say it's relatively easy to collect the ants once you're shown and get the hang of it, but it's even harder to get the fungal cultivar, the pathogen, and the bacterial mutualist all into what we call pure cultures, and then also keep them in pure culture. Just a little bit of humor. I like to add that I conducted some additional host parasite studies by infecting myself, not on purpose, with botfly, trematodes, some ant kin recognition studies with bullet ants and other risks of the field. It's not all tropical fun. And then again, I perform these bioassays, also including the Escovopsis pathogen, but instead of the cultivar here, using the symbiotic pseudonocardia mutualist. And so again, just here's an example of a pseudonocardia that's doing a good job of inhibiting the pathogen. And then here is an example where the pathogen doesn't really care about this strain and basically ignores it. So in my initial collecting and culturing of these microbes, I found that the cultivars are not necessarily facing one particular strain of this Escovopsis. They're actually facing predominantly 3 different strains. I kind of refer to them as morphotypes or phylotypes. We genotype them and they are genetically distinct, so they're not just, sometimes they'll change color or do different things in culture, but they are genetically distinct. And I was a little bit discouraged by, finding these various strains, in part because, the history of coevolution kind of starts with the dichotomy of parsing out interactions to highly specific and co-adapting versus diffuse associations that are sort of less likely to involve a specific pairwise coevolution. But at the same time, I was reading this book called The Geographic Mosaic of Coevolution, which in many ways is sort of a broad framework for approaching and describing coevolution and the coevolutionary process. But I also kind of gravitated towards this book because it starts with, this quote, but there's much more to the evolving web of life than the artificial dichotomy between local pairwise reciprocal selection and diffuse coevolution. And it sort of gives you a way to approach these diffuse associations and sort of tease them apart and think about, okay, maybe there actually is a highly specific co-evolved interaction amongst these diffuse interactions. So one of the first things I did was simply compare the average zone of inhibition across the different strains. So that's what you're looking at here. How much inhibition was there across these different strains? And so what we find is that there's, even though we just have 3 points, but there is a nice sort of correlation here where we're encountering this brown strain mostly in the environment. It's the most abundant one. And then this yellow and this fuzzy brown are occurring at a much lower frequency. And so I don't think it's a coincidence that the inhibition is the greatest to this brown strain. And that's sort of consistent with an idea we call parasite rare morph advantage, simply meaning that the more opportunities you have to combat something, you're going to basically evolve to do it just through exposure. So that was kind of nice. So what we can now do is say, okay, well, there seems to be, this kind of rare morph advantage going on with these two strains. And then what I want to do moving forward is look at this interaction between the bacteria, the pathogen, and say, okay, can we actually find this example of local reciprocal adaptation, within this framework of the geographic mosaic of coevolution? And so what I'm doing now is kind of digging down into whether or not we can pick apart local adaptation and these mosaic patterns within only the brown strain and the Pseudonocardia. So what you're looking at here is a phylogeny of the Pseudonocardia strains, again, across Costa Rica and Panama. And one thing I'll point out, a couple things I'll point out. One is that you can probably see by your eye, but we do have, what we call phylogeographic signal, or we have a pattern of isolation by distance. And that's important because if, going back to this acquisition hypothesis, if the ants are pulling up, horizontally soil microbes from the environment and using them, we don't expect to see this phylogeographic isolation by distance pattern. This pattern is more consistent with an animal that has limited dispersal, and it's probably a reflection of being, tied to its ant host. And then the other thing I'll point out is you'll see, particularly here within Barrow, Colorado Island and Gamboa Forest, these really high, what we call high identity sequence clusters. So there's basically almost no variation in these clusters. Why that's interesting is because it's often touted that when you see these high identity sequence clusters, it's basically a sign that the population has gone through a bout of purifying or periodic selection. And so this is just kind of a model of what that looks like, and that's very similar to what we're seeing in Gamboa and BCI. One thing I'll point out, again, if we want to look for a signal of local adaptation or reciprocal adaptation, basically, theory tells us that when we have congruent population structure, that we're more likely to see that. So this is just kind of a graphical representation of that. Imagine a host and parasite and having similar population genetic structure, you're more likely to see local adaptation arise there. Whereas, if there's asymmetries, you're going to lose the capacity to form, local reciprocal adaptation. And so to basically test whether or not this is happening, just ran a correlation between the population genetic structure using FST analogs between the fungal pathogen and the bacterial mutualist. And we do actually find, a loose, but albeit a correlation enough that shows there is a degree that they're tied together, right? If we didn't see any correlation, we probably wouldn't predict finding any signal of local adaptation. Another thing that I looked at in these bacteria is whether or the degree to which they're recombining. So, we often sort of assume that microbes are clonal, and they largely are, but what can actually happen is nature is you can have, bacteria that are highly clonal, ranging from highly recombining, so much so that they actually, the recombination rates resemble a sexual population. And so, if we are, again, looking at this sort of acquisition horizontally versus vertical transmission, if it's going vertically, clonally through the host, we sort of anticipate to land more on the clonal side, whereas if they're, using regular soil microbes, we expect, recombination rates that basically mirror what we naturally see. And so I tested that here. Long story short, we're basically falling on being highly clonal, which again is sort of suggesting that there is verticality and specificity with the host. This is just kind of a graphical representation of what that looks like. I'll say at the time that I did this, these recombination rates were amongst the most clonal rates ever found in bacteria. But this is just a graphical representation of what it looks like when you do see recombination events. And we did find a few recombination events. So just to kind of summarize that, within the pseudocardia, we do have phylogeographic structure, isolation by distance, we're largely clonal, and we're congruent with our Escovopsis pathogen. So we've checked off some boxes that we can now say, okay, there's a chance we can actually find some local adaptation here. And I should have said this earlier, but the phylogeny that I showed you is between 70 or 80 samples, and it's I used 5 housekeeping genes that are sort of commonly used to quantify those recombination rates. But I'll say in a genomics paper, we went back and fully sequenced about 30 subset, and the population structure looks exactly the same. Although I'll talk about some of the genomic differences here in a minute. So what you're looking at right here is, again, these zone of inhibitions, and here are the locations of the pseudocardia. So again, we're looking for, going to try to look for a signature of local adaptation within this data set. But I'll point out a couple different things. This again is our focal populations here in Panama. And this is kind of the phylogeny for context. And like I said before, when we see these high identity sequence clusters, that's typically taken as a sign of purifying selection. And I don't think it's a coincidence that where we see these the most in BCI and in Gamboa, the most cohesive units, that's where we're seeing the highest inhibition by the mutualist. Interestingly, for some reason, when you pair, the La Selva, Costa Rica pseudocardia, they are basically not doing as well against this particular pathogen. And in some ways, that's a kind of a cool finding too, because it's consistent with some of the predictions in the geographic mosaic theory about seeing selection mosaics over space. And we're more likely to find that local adaptation where the selection mosaics are the most intense, which in this case, would be Panama versus Costa Rica. So what you're looking at right here is what's called a reciprocal cross test. And it's basically just a standard way to look for a signature of local adaptation. So on the Y is the zone of inhibition, and then on the X is the pairings between the pseudocardia mutualist and the Escovopsis pathogen. And basically where you see the color coding, where the population locations are paired, if that is the highest amount of inhibition, that basically means that you detected a signature of local adaptation. So just moving through it, we see this nice clear signature here. Good result here on Barrio Colorado Island. When we go to Pipeline Road, we have no signal whatsoever of local adaptation. And then when we look at Gamboa Forest, it's kind of somewhere in between. They're really good at inhibiting their local population, which is a good thing. Not so much compared Gamboa to Pipeline Road. But then there's also this, equally as high interaction between Gamboa and BCI. Just for a little bit of context again, I'll remind you that we predict the local adaptation where population structure is most congruent. This assay right here, or this test right here, is just another way of presenting population structure. This FK is a signal of how derived the population is. And so, for both of the players here, Asclepius and Symbioncardia, they're sort of the most derived within BCI. And again, I don't think it's a coincidence that that is also where we're seeing the highest, or rather the real signature of local adaptation. This again is the same pop structure data, just presented a little differently, but basically it's a visualization of kind of what I was just saying, where you see the most overlap between Symbioncardia and Asclepius and BCI, is where you see the strongest signature of local adaptation, little congruence in Pipeline Road, less in Gamboa. So to just kind of summarize that, we can now take this supposedly diffuse association, we can parse off the two rarer strains into rare morph advantage. And yes, we can say that we found local adaptation and we had some predictions consistent with our GMC model. Again, local adaptation was not perfect. We didn't find it, consistently across the three focal populations, but it makes sense when we put that against our population genetic structure congruence. So to bring us back to, where this fits in with these potential, transmission mechanisms, it's clear that we have, vertical transmission. Like I said, we don't necessarily see these other horizontal transmissions, but we can kind of rule out this initial acquisition of microbes. But we have answered this verticality aspect, but we haven't really done is answer, well, how is the potency maintained, right? We talked about some of the dangers of clonality. And so I'll just briefly talk a little bit about some of the genomics that we did that speak to this. So what you're looking at right here is a chart of what are called BGCs, biosynthetic gene clusters. And so these are the regions of the genome that we predict are producing the secondary metabolites or the antibiotic molecules within the symbiont. Like I mentioned before, this comes from the subset of the 30 genomes that we sequence. And what's interesting about that is what you're looking at here in Gamboa, Pipeline Road, and BCI is basically the accumulation of novel biosynthetic gene clusters. They're loosely grouped into different families, but each dot represents a unique biosynthetic gene cluster. And what's really, I think, quite interesting is that, in both Gamboa and Pipeline Road, which, if you remember from the big phylogeny, have a huge amount of genetic divergence and genetic diversity, there's really only a handful of these unique biosynthetic gene clusters. By contrast, if you look at BCI, and remember that phylogeny has really high identity sequence clusters, so there's very little genetic variation in what we would call the core genome. But by contrast, here in BCI, we have this accumulation of biosynthetic gene clusters. And so what that tells us, and what we sort of proposed in our model of how this system is evolving and maintaining potency, is that, bacteria have these other mechanisms to acquire genetic material through lateral gene transfer. In this case, many bacteria have a plasmid, which is not considered part of the core genome, but is more loosely associated. And so in our genomic work, we found a signature of a plasmid. We're sort of finding that these are residing on those plasmids. And we did a lot of statistical analysis to see if we could specifically tie any particular gene cluster to the local adaptation signatures in the inhibition assays. And the long story short was that we couldn't just tie a particular cluster to the local adaptation signature. So it's not that you just kind of are picking up these things and then they're sort of making quick use of them, but rather, once we scanned within these clusters for signatures of selection, trying to tie it back to those bouts of periodic selection, then we can actually start to explain the bioassay patterns. So it's kind of a two-step model whereby, there's vertical transmission, the core genome is maintained, There's the acquisition of these BGCs, probably laterally through the environment. But then there has to be kind of a bout of passing through a periodic selection to, get these, to give us the signature of local adaptation. So that's sort of the main big story I wanted to tell you about the fungus farming ants. I'm happy to talk more about the specifics of some of the genomic work that we did. And so now I'm going to make a pivot here, and hopefully it's not too harsh, and I'll keep you along, but just sort of a show of hands, I've talked about this incredible ant symbiosis. We know a lot about it. It's one of the best studied symbioses. We know a lot about other great, fantastic symbioses like bobtail squid and Vibrio. And, it's easy to be like wowed with their complexity and feel like, wow, these are just like such rare things. But as a show of hands, do you think that these symbionts in nature are rare, or are they common? So do you think they're rare or common? Yeah, so I'm with you. I kind of think that they're common as well. And I don't think it's a coincidence that we know a lot about fungus farming ant symbiosis. We know a lot about Vibrio symbiosis, in part because we can see with cultivar, we can see the bacteria, although, it might be hard. We can see it without a microscope. We can culture the pathogen. We can culture all of the players. And so that's basically why I think we know a lot about it. Same goes for Vibrio. You can culture all of the players. They have this bioluminescence that just screaming, look at me. But, from our microbiology, we know that most of the microbes in the world we cannot see and we cannot culture. So when I came to UCLA and I started working in Paul Barber's lab, I became involved in a project looking at marine microbes. And basically, it was a big multi-institution project describing basically the metazoan populations in coral reefs in Indonesia, in the Coral Triangle. And there was this big data set of all of the animals associated, but also 16S microbial profiling of the bacterial associates of the different organisms. And so I was really interested in this data set in part because I feel like it kind of gave a rare opportunity to address, sort of some of the commonality of or how common these symbionts are. And so, if you're in the symbiosis field, one, the main questions we're always asking is, is your microbiome composed of primarily tourists or residents? We think of things as, are they symbionts or are they transient? And so I was sort of interested in this, exploring this question within this particular data set. So this is the Coral Triangle. And basically the way this study works is that there are these structures called ARMs, which are autonomous reef monitoring structures. And so they basically get buried next to the reef. They stay there for 3 years and they get pulled out. They look like this. And basically there's a really intense effort to categorize and genotype everything that's there, from the animals down to the microbes. And I was interested in, addressing, again, can we find signatures of symbionts within the system? And the way that I, approached it initially was just through a very simple test. So, in the fungus farming ant stuff, you saw me use a lot of, are we seeing this isolation by distance pattern that is mimicking the animal host, or are we seeing, ubiquitous microbes and what someone call everything is everywhere hypothesis? So I basically took similar approach to this data set, but what's happening is we're going up in scale in 2 ways. One, we're going up in scale geographically. Now we're talking, a much greater distance in thousands of kilometers. And we're also going up in scale. So rather than looking at individual lineages or species and their geographic patterns, now we're looking at community similarity. So we're not just using one lineage, genotyping it, we're just doing a community profile. So whereas we were previously using FST and population genetic structure, what we're using here is basically community similarity. So this is just kind of a graphical representation of, if these, bacteria associated with these animals are, just transient, just part of the seawater accumulating, then we don't expect to see any kind of isolation by distance pattern. However, if we do have, specificity in these microbes having population structure structured by host dynamics, and again, from this data set, we know that the animal hosts have geographic, isolation in their community structure, then we expect more of basically a diagonal pattern. And long story short, that's basically what we find. You'll notice that the diagonal is going the other way, and that's just simply because here I'm using community similarity as my mark, and then in the program that we use, CHIME, they use community dissimilarity. So that's why it's just going the other way. I'll just mention that when these arm structures are pulled, they're sieved into different fragments so that the metazoans fall out into different categories by their size. And basically it didn't matter which size fragment we used, we got a similar pattern of community isolation by distance, which again tells us that probably the majority of these bacteria that are sitting on these metazoans are probably having some intimate symbiotic role because their host population structure is pulling the communities along with it. I'll kind of button things up here real quick. I just want to mention, since I started here at UCLA, I've started a mentorship program called Marine Science Pathways, where I work with undergraduates, both here at UCLA, but also from surrounding community colleges, and specifically underrepresented students in the Los Angeles area. And so this little project was with 2 of those students, having them put this figure together. One last thing I'll just mention, where my research is taking me currently is into this really cool isopod system, which again is a marine habitat, but actually has a lot of parallels with the fungus farming ants. So this is an isopod that has episymbionts, cyanobacteria growing on top of it. And it's again having a dual function in that it's like the ants is serving as their primary food source. They eat the cyanobacteria. They're seeking out sunlit spots in specific locations to grow up the food source. But then there's another function whereby there is the cyanobacteria are producing some kind of, again, secondary metabolite, some kind of molecule that is serving as defense, making them basically unpalatable to fish. So what's cool about this system is it's really set up for some cool ecological switch experiments and comparative genomics, in that we have lineages that have the symbionts, some that don't, have some that have the symbiont as a nutrition source, but not as a defense source. And so we're sort of moving into exploring a lot of similar questions about transmission and specificity with the ants in this isopod system. So with that, I'll take questions. And this is my PhD advisor, Cameron Curry, looking sad and sunburnt on a pecky on the way back from a Los Amigos station in Peru. Thank you. Yeah.
Behavioral adaptation from the ants to insect teaching?
Yes. I'll say when I started research as an undergrad, it was in the fungus farming ants, and it was all behavior. I was like convinced I was going to be a behaviorist. So the bacteria isn't doing all of the heavy lifting. So the ants can incredibly, they can sense and pick up and remove a single Escovopsis spore. And so the way it works is The infection is typically at the bottom of the colony, and where the fungus crop grows is through the top, so they typically add on the top, so that it's sort of a chase thing where the pathogen's always coming up. And basically, if it starts to get too high, the ants will just ditch like half of the garden and then just take it out and throw it away. And there's also grooming behaviors specifically that they do on each other if they have the spores. And the fungus in the large leaf cutters where there's like extreme caste systems, like there are like undertakers who all they do is move dead bodies around, and they're once they become an undertaker, they're not allowed to go back into the colony because they'll expose the colony. So yeah, there are really sophisticated behaviors. So if one chamber, you know, is overrun with infection, they'll close it off to save the rest of the colony. So yeah, they do a lot of incredible stuff. Yeah. And also with the grooming behaviors, like they specifically are grabbing the bacteria and moving it and stuff. So, yeah.
Do you think there's any kind of effect of the material that they're gathering? Like any changes in the plants they gather from the fungus farm?
So that's a great question. So in the leaf cutters, they, again, like extreme caste system, they have one caste that they just call hitchhikers, and they literally just like sit on the leaf while a larger worker is carrying the leaf. And so one of the things they do behaviorally is they swat off like parasitoid flies that may infect the ants. But the other thing they do is they actually start cleaning the leaf and removing microbes, so that by the time it gets to the garden, it's like a lot of the microbes have been removed. What's pretty incredible about the ants is that the leaf cutters is that they can harvest like 30% of the plant material in the rainforest. So if you know anything about diversity in the rainforest, like 30% is a huge amount of species. So it's pretty incredible that they can take really tough stuff, really tender stuff, and from a huge diversity. And part of that is because they are manipulating it before they feed it to the fungus garden.
Do we see like similar kind of spread of different types of these bacteria in the isopods like we do with the fungus, or like locally we have very
that's a good question. So we're working on that right now. So we're specifically genotyping the cyanobacteria to look at that. We know that just like the ants, there is, even though the isopods are in a fluid environment, they are pretty cohesive. There's well-defined population structure, just like there is in the ants. So that's kind of one question I'm working on right now is on two levels. The specific symbiont strains, are they different across the populations? And then also at the community level, are there different consortia across the different populations? So I don't know, but I'm working on it. Yeah.
Regarding the work you're doing on the microbial communities, I was wondering if there's a way that you can actually parse that data set to try to distinguish whether or not the associations are driven by symbiosis versus other types of interactions that might be specific in the bacteria and the protozoa.
That's a good question. I basically, to dig into those questions, you need a species-specific system. So I'm sort of interested in those dynamics in the isopod system, but in that broader data set, I mean, it's just kind of coarse, like 16S profiling with huge data set. So it's a little bit correlative. Like we can't really get to mechanisms and things there, but yeah, I know what you mean. Yeah.
You commented on that human taxis. How does it work? I can understand like it's in a mobile base, but is it the sport?
No, it's the hyphae. So like, I don't think I have any images, but basically, you can do these, just like the bioassays I showed you, you do something similar, but instead of giving it like one, either the bacteria or the fungal cultivar, give it like multiple ones. And like you can see it'll like, it's pretty incredible because a lot of these ant species are sympatric. They're like right on top of each other, but you can see that like it's the hyphae is just darting directly for the cultivar that its species is used to, and it's just avoiding the others. So I don't quite understand the chemistry of the mechanism, but it's crazy when you see it because it's just like doop, and then once they've attached with the hyphae, then they'll start devouring it and the spores will come out. But yeah.
Thank you for an awesome talk.
Thank you.